Before starting, I would like to thank all the organizers for the very nice opportunity. So as you can see, the title is quite broad. And that's because I would say since I was PhD students, I've been working on different projects related to tumor invasion, and majority of them at least were done within different multi-scale framework. So just I'm going to start with a little bit of like more general introduction, but then the results I'm going to show you actually comes from a recent work I've been done with in collaboration with Giulia Chiari and Marcello Delitala from Polytechnic of Turin. So we already have seen this picture quite a lot in these days. I'm not going to go into details, and already Thomas nicely explained to us all of the hallmarks. But the one I'm more interested in, at least for this talk, but in most of my projects are these two here at the bottom. So of course, as the title said, tumor invasion on one side, but as we said, all the hallmarks are connected. And in particular, angiogenesis is highly connected, is strongly connected with tumor invasion. The mechanisms that can drive invasion or angiogenesis are multiple. There are several mechanisms, several processes. I listed here just some of them, just for like some of the one I've been working on. For instance, invasion can be driven, or is surely driven by the receptors located on the membrane of the cells, as well as the fibrous orientation has a role in the way cells, like tumor cells, invade the tissue, the ECM stiffness at the same time, or as we also heard this morning, the acidic of the environment could drive as actually the way cells invade the tissue. At the same time, for angiogenesis, and we heard about it several times, oxygen plays a very important central role in leading the angiogenic process, as well as the VEGF production by tumor cells, or the interaction between tumor cell and endothelial cells. But among them for today, the two main elements I would like to focus on are these two. So I'm gonna focus on the way oxygen level and oxygen variability can drive invasion through the action of a specific protein that actually the cells produce, and that allow them to respond to different level of oxygen. So this protein is called actually snail. So in general, in healthy and cancer cells, snail has been shown as to be a key inducer for migration, and in particular is highly related to the EMP transition that we also talk about a little bit these days. In particular, when we are in hypoxic condition, snail is highly expressed by the cells. So if you look here in the table on the top right, these are the 4 different breast cancer cell lines exposed to 0.1% of oxygen. And as you can see, snail is overexpressed in all of them. And if we also look at the spatial distribution of the protein within the tumor mass when it is exposed to gradient of hypoxia, as is shown here, the gradient of hypoxia go from the outer border toward the inner border. So here the oxygen is lower, here the oxygen is higher, and the brownish cells are the ones showing the higher level of snail. So as you can see, snail is highly expressed here at the inner border, while it's lower expressed in the outer border. Moreover, there are also other studies that then look at the way upregulation or silencing or downregulation of this the gene, and consequently the protein, can affect the way cells migrate. And for instance, here at the bottom, we have 2 examples, again, some different breast cancer cell line. And when snail is upregulated, this causes increase in the way cells migrate, and therefore increase in metastasis and increase in the poor prognosis of the patient. Instead, if this gene and consequently the protein is silenced or just downregulated, what we can observe is a reduction in the invasiveness of the cells, so a reduction in the way the capability of tumor cells to invade the tissue, and therefore a more localized and confined tumor mass. So these are the main biological ingredients. Again, using a kind of bottom-up approach, we are going to use this to build up our setting. That is going to be, as the title said, a multi-scale one. So how we build up the model is considering these 4 different levels. So we have a microscopic scale at which we describe a single cell's dynamics. We move then to the mesoscopic scale, in which actually what we can give is a statistical description of the population of cells themselves. And then there are proper techniques that allow us to go from the mesoscale to the macroscopic level. There is the one that then we are going to simulate, in which we observe the collective behavior of the tumor cells. And one of the the interesting aspect of doing this multiscale modeling is then we can connect, or we can observe the way the macroscopic parameters that we define at the microscopic level are connected directly to the macroscopic parameters of our model. So instead of stating the way macroscopically cells move, their movement, because we're interested in the invasion, will be directly driven by the macroscopic parameters in a very interesting way that depends on the way the scaling is done. And just as a remark, the aim of this study was actually to understand the interplay between the snail protein and oxygen or hypoxic condition in driving the invasion or the migration of the cells. So briefly going to the model, our population is statistically described using a distribution function P that depends on 4 microscopic variables. We have time, we have position of cells, microscopic velocity, and then we have an additional variable Y that for our case, it describes the snail expression, so the expression of the protein into the cells. Of course, there's a very flexible framework. It has already been used several times in several different contexts, and this variable can be used to describe, of course, other elements, like for instance, membrane receptors. But for us, as I said, is snail expression. So how we are going to describe at the microscopic level how XV and Y vary. So for Y, our snail expression, what we model is basically a production of our protein that depends on a self-inhibitory mechanism, because snail self-inhibit itself, and it depends also on this function H of the oxygen. So there is, of course, a dependence on the production of this protein, or the oxygen level influence the amount of protein that is produced by the cells, and in particular, as we said before, the higher the oxygen, the lower snail. As a remark, for this study, the oxygen is fixed in time. So we have a fixed distribution of oxygen. We want to see how cells respond to it. But of course, as next step, we are thinking about extending this to a varying oxygen that is much more realistic. So we have our production part and degradation part. For the velocity of these single cells, what we model are simple acceleration and deceleration of cells. So the acceleration is kind of natural, while the acceleration part is described by the function G. And what this function G include are 2 different mechanisms. There is a chemotactic force driving cells to move towards increasing gradient of oxygen, and there is an anti-crowding mechanism. That means if I'm in an area too packed, too dense, there is too many cells, I'm trying to move far away from it. As you can see, there is also a dependency on Y in this acceleration, and that's because snails, as we saw before also from the data, tends to increase the motility of the cells, and therefore the velocity at which they move, in this case the acceleration, would depend on the level of the protein. And finally, for the position, simply kinematic law describes the way the cells change their position. So these 3 equations would represent our microscopic system, and what we do is to consider a classical kinetic approach. So we build up the kinetic transport equation that describe the way the distribution function P for the cells evolve. And that's classically done. On the right-hand side, what we have are the gradient with respect to the 3 macroscopic variables and the 3 fluxes we just build up for the function G, and that's what I just showed you before. And instead, on the right-hand side, we just consider proliferation of cells. So since the talk is going to be more on invasion, I'm just going to show you the general expression for our proliferative terms. We did some study on it, if anybody's interested, because also snail has a role in growth. So we have done some analysis, but the majority of it was on invasion. Just to give you an idea how P is built, because also connected with what we said on Monday with Thomas. So P, our proliferative operators, take into account 2 elements. So we have a first function mu1, that it can depend, as we said, on several factors, because proliferation can depend on the adjacent, like on the oxygen, on the nutrients. It can depend on the amount of macroscopic density of cells themselves that I have in a certain point. It can also depend on the speed of the cells, because if we, for instance, consider the go or grow hypothesis, or a general trade-off between go or grow, the faster I move, the less I'm able to proliferate. So all these influences can be included into mu1. Instead, we consider an integral terms to describe the influence of snail into proliferation. So mu2 of y would represent our function that take into account the fact that the higher the snail expression, the lower the proliferative capability of the cells is. And this is a way to basically describe how the level of the daughter cell will be related to the level of the mother cells, depending on the assumption that one can make on the kernel key. Okay, so that's the mesoscopic level. There's a very classical theory of kinetic models that allow you to derive from the kinetic equation the corresponding macroscopic system. So it is based just on like rescaling the space and time. And what we assume is that the microscopic processes related to snail production and degradation are much faster than all the other processes related to cell development, so cell progression. Assuming this rescaling over here, using the classical method based on the definition of the moments of P, the moments equation, and then the expansion of the moment, what one can obtain formally in the limit when epsilon tend to 0 is this macroscopic system over here. So this is a system for the evolution of M0, that's the macroscopic density of tumor cells, and M0Y. M0Y represent the macroscopic average expression of snail into the population. So it is very interesting to observe that the equation for M0 has basically all this transfer part, drift part, and in particular, tactic sensitivity of this drift is related to this function f. So if you see the expression of f directly take into account all the microscopic parameters that we have modeled at the microscopic level. So it take into account transcription rate, degradation rate, it take into account the function h that describe the inhibitory mechanism of oxygen on snail, it take into account acceleration and deceleration. So it directly comes from the way macroscopically single cells move. And that's what I was saying before. So that's one of the good aspect of multiscale modeling. So we do not state the expression for our tactile sensitivity, but it comes directly from the way single cells are described. And as you can see, it's pretty a nonlinear function of this coefficient. And another interesting part is that f also comes into the expression of M0y. So if you just write this expression differently, actually you can substitute f times M0 with A1 over A2 times M0y. What does it mean? It means that the drift is driven, the drift in the tumor cell population is driven by the average expression of snail into the population. So this is the model we actually simulate. I would like to show you two numerical examples. So in particular related to model validation and comparison with data. We also did more qualitative analysis, but let's stay on these two for also time reasons. The first one relates to, so what we tried to do was to replicate some experimental data, the one I show you initially. So we wanted to replicate the data that shown how hypoxia or snail control and tune can change the capability of cells to move. So the experimental setting basically is made up of two different chambers. So I think that some of you at least are familiar with it. So we have two chambers. Cells are located in the upper chamber. There is a membrane between, and they are allowed to cross the membrane following a certain gradient of nutrients. And what the experimentalist evaluated is at a fixed time, how many cells that cross the membrane in different conditions. So we have two different cancer cell lines. We have adenocarcinoma cell and breast cancer cells. And these are the data for breast cancer cells. So the black bar are the data. So I just take them from the paper that was cited before. And what they do were experiment on varying snail expression at fixed and normoxic oxygen condition. So we have a control with normal oxygen, normal snail, and then we have the scenario B and C, where we have overexpression of the protein or knockdown of the gene related to the protein. So how we try to replicate it? So if we look here at the picture on the left, so here what we are plotting is the function f as a function of GS. There is the transcription rate of the protein and the oxygen. So for our scenarios, we are keeping the oxygen fixed and we are changing the level of GS. We are just tuning this single parameter. So how much basically snails can be produced by the cells. So for upregulation, so our control is A, sorry for red over red, but for the upregulation, we use the value B, and for downregulation, we use C. And as you can see from the results, the red bar are our modeling results. So we use same time frame and spatial frame of the experiment, and we were able to capture the behavior that was shown experimentally, meaning that when I overexpress nail, I basically almost double the capability of cells to move. So in the same time window, double of the cells have crossed our membrane. Instead, when I knock down for nail, cells are not able anymore to move, and we can see this decrease in the fold change in cell migration. A similar experiment were done for adenocarcinoma cells, but in this case, we have again a control that normoxia plus normal Snell condition. But then we have scenario D, there's hypoxia, just hypoxia, same Snell condition, and scenario E, that actually is hypoxia plus silencing of the gene. What does happen? So as you can see, again, black is the data, red is the model. So we have A as before. Now for D, we simply decrease oxygen, keeping the same level of the transcription rate. And instead for E, we take the same value that we had here before in what? Okay. It wasn't expected. There. Okay. I'm sorry. So I just was saying that for the silencing, we take the same value we had before in C and just keep it for the E scenario. So yes, thanks. I don't know what happened. Yeah, so E is same GS, but just lower oxygen. So as you can see, when we have just hypoxia, we expect and we observe increase in cell migration. But when on the same oxygen conditions, snail was silencing, we went back. Actually, we decreased the fold change in cell migration with respect to the control. And it's nice to see how the model actually captured pretty well the qualitative but also quantitative behavior of the data. Just a second test, and then I'll let you go for coffee. We also want to observe if we were able to capture the spatial distribution of snail, not just the changes that snail allow cells to make in their migration. So this is the initial condition of the model. Now we have, again, to the setting. Cells are located here at the center with a nice Gaussian distribution, while oxygen now is again fixed, but higher at the border and lower in the middle. So if we have a 1D, like if we just consider the 1D cross-section, we will see the continuous line for the tumor cells and the dashed one for the oxygen. If we let the model evolve just the way it is, without touching anything else, what we would get is for the tumor cells here at the top row, since cells are moving towards the oxygen and they try to avoid crowded area, the two mechanisms work together and they just spread around the domain, like far away from the center, and these ring structures start to emerge as we would expect. But interesting is that if you look at the distribution of MY, that remember is the macroscopic average distribution of snail into the population, what you would see is that snails stay higher in the area where oxygen stay lower. It still have the same like ring structure because we are evaluating snail into the population, not the extracellular one, but it actually is able to capture nicely this higher distribution of snail into the inner border and this lower distribution into the outer border. Okay, so just to wrap up what we said, so the key finding, or at least the key element we were able to get from this model is that we were able to observe what experimentally have been observed. So this interplay between hypoxia and snail, and also like play a little bit with them to see how it's possible to tune one of the other in order to control a little bit the response of tumor cells to them. And in particular, the comparison with the data and with the up and down regulation of the protein was very interesting because allowed us to show room for possible improvement of the control of snail also as a therapeutic target. Actually, I read something. There is already some therapies that look at this specific protein, but I think that there is still a lot of work that can be done in that direction. And as I already said, this very simplified, just starting from oxygen is fixed. It shouldn't be. It should vary. And we are right now start to to see how we actually include it. And also another actually what's next that I would have included, I just forgot, is work deeply into this EMT transition. So try to see if we can actually catch also this. So with this, I would like to thank for listening and thank also like the funding for letting me be here.